Thank you for joining me on this last video of our module. And while in the previous video we talked about cell division of all cells in the body with the exception of the sex cells, on this video we will be focusing on sperm and egg cells. So this video is going to be all about the meiosis. And as we remember, meiosis differs a little bit about the division of all other cells, because when we make sex cells, sperm and egg, we only want half of the genetic material within them. This was of course because when we have sperm, which contains 50% of the genes, and an egg, which contains 50%, well, when they fuse together at fertilization, we get a new individual that has the full 100% of the genetic material. And in the proper language of the genetics, we use rather a lowercase n for symbolizing each 50%. And the term for this is haploid. So N from the sperm and the N from the egg cell, when added together, that makes 2N. And that's for the new individual. So 100% of the genetic material is 2N, and that is called diploid. And before we look at the process of meiosis, one step by step, we will need to review a few important processes that apply only to meiosis. And we did not see them before when we talked about mitosis. So we will be looking at the synapses and the crossing over. Let's start with the synapses. What happens here is that the chromosomes, which would not otherwise connect to each other, they pair up right next to each other. So this may be so that there is even a little bit overlap between the two. So that is our synapses. And the other concept, which actually relates to this, is known as crossing over. In this one, the chromosomes that have parts overlapping each other, they end up exchanging parts with each other. And this does not happen just in one spot at the one time, but this can be multiple spots being exchanged at once. We just tend to show only one to make it easier to follow. So that's what happens in synapses and crossing over. I hope that it makes sense, as these are two fundamental concepts that we need to know as we start to look at the process of meiosis in more detail. So next we will look through the stages of the meiosis. Let's do that. And unlike with mitosis, there is going to be two cycles of cell division that take place. Therefore, we have here as the first step prophase 1. And all of the things that we are familiar with are included. Nucleus and nuclear envelope around it disappears at this stage. But instead, the structures that we already know, the centrosomes and spindle, they both appear, so become visible. So those were events that we are already familiar from the prophase in mitosis. But since this is meiosis, we have a little bit more going on here. So during prophase 1 in meiosis, we get the chromosome pairs to form synapses. And as you remember, this referred to them pairing up next to each other. We also have some crossing over happening. In this, parts of the chromosome pairs are exchanged between them. So both of these events, forming a synapsis and doing crossing over, they both contribute to mixing up the genetic material. And the resulting new mixed chromosomes are made. Just for the sake of keeping the diagrams manageable, I will not show the results of crossing over on this description on these figures. So now we are ready to look at the metaphase 1. In this stage, similar to what we saw in mitosis, now the chromosomes, they line up on the equatorial plane. But the difference is that they line up in pairs. Now, during anaphase, these chromosome pairs separate as they are pulled to the different poles of the cells. And we also see the cleavage, so the pinching off into two cells starting to happen. And now we are ready to look at the last stage of the first cell division cycle. And this is rather unsurprisingly called telophase 1. Here we see that the nucleus and nuclear envelope, they both appear again. And the original cell has divided into two cells. So that was our first round of divisions in meiosis. But we are not finished just yet. In meiosis, we go through the second round of divisions as we want to reduce the amount of genetic material from 100% to 50%, both in sperm and egg. So let's do just that and continue where we finished earlier. And we finished with the first round of division with the telophase 1. Let's continue from that. And now during the second round of meiotic division, we start with prophase 2. Just like before, the nucleus and nuclear envelope disappear and the centrosomes and spindles become visible. Let's look at the next stage known as metaphase 2. So just like in metaphase in mitosis, the same happens here in metaphase 2 of the meiosis. We see the chromosomes lining up on the equatorial plane. This is followed by anaphase 2, and during this, the chromatids separate and they are pulled to the different ends of the cell. And we also see the cleavage starting to form. And the final stage is, of course, telophase 2. This is characterized by the nucleus and the nuclear envelopes becoming visible again. In addition, we have, of course, now 4 new cells. So that completes the process of meiosis. And here I have listed all 8 stages of meiosis. And as I'm sure that you notice, there are these 2 cycles of division that take place, so we are able to halve the amount of genetic material. Please also notice that there are the synapses, which was the chromosomes pairing up next to each other, and the crossing over, where parts of the chromosomes exchange material. Both of these aim for introducing more variation to the final product, the resulting 4 cells. What is interesting here, I think, and really important to note, is that the resulting 4 cells are all different. So they all have a different mix of DNA in them. This is how we have variation among the offspring. You never know what kind of characteristics the baby is going to inherit from father and mother. The mix can be a lot of different options. Now that we have looked at the meiosis, I think that it would be a good time for us to compare that with mitosis, which we had a look in our earlier video. And we remember that mitosis, which we looked in earlier video, was what happens in everywhere in the body with the exception of the sperm and female egg. And meiosis instead was the special cell division that happened only with gametes, so only our sex cells, the sperm and the female egg. Let's start by noticing that while in mitosis there was one full cell cycle, which results then of course in two cells. And we call these daughter cells, the cells that result from cell division process, the final output. Well, in comparison in meiosis, we will have two cycles. Remember, we had prophase 1, metaphase 1, anaphase 1, and telophase 1, followed by prophase 2, metaphase 2, anaphase 2, and telophase 2. And because of this, the first division resulted in two cells, and then they divide again during the second cycle, producing in total at the end 4 daughter cells. Great, let's see what else we can tell. And because the DNA content was duplicated before the cell division in mitosis, we end up with daughter cells that contain the full 100% of the genetic material. These cells are known as 2N or as being diploid. Although meiosis started the same, there is no DNA duplication before the second round of divisions. Therefore, this second round halves the amount of DNA present, and we end up at the very end with daughter cells that have 50% of the entire DNA content. So those are some big differences there, right? Let's look at the final one. Finally, I want to note that the mitosis resulted in genetically identical daughter cells, 2 of them. So these cells were exact copies of each other in terms of their DNA. While instead, on meiosis, we are left with 4 daughter cells. Remember, the first round produced 2, and the second round divided these 2 again. And all of these daughter cells have a unique mixture of DNA. So the resulting daughter cells are all different from each other. And they are, of course, also different from their parent cells. So I hope that that clarified a little bit about the similarities and differences between mitosis and meiosis. I want to finish off this video by looking at a real-world chromosomes a little bit more. And a technique for visualizing the chromosomes of an individual is known as karyotyping. In this technique, we visualize all 23 chromosomes of an individual. And truth be told, they often look like this bundle here in the figure, but we do often go and organize them in the order of the size from the largest to the smallest, looking like this. And that gives us a nicely ordered diagram, but also helps us to identify the chromosome number for each. And what we normally do from karyotype is that we study it to determine, for example, if there is the right number of chromosomes present. So we should see 23. We also look at the size of the chromosomes. Each chromosome should have symmetrical chromatids. And there should be nothing missing or extra in any of these chromosomes. If there are any of these issues, it is an indication of a genetic disorder. And there is one more thing that the parents usually want to see, and that is, of course, the sex of the child. If it's a boy, which would be XY, or a girl, which is XX. And this completes our final lecture video on mitosis and meiosis. I hope that you have found it useful. I would especially encourage you to know what happens at each stage of both of these, and also how they compare and contrast between each other. So the table that compared these comes handy too. Thank you for having joined me on these videos.